Hello, everybody. Welcome to this virtual symposium on multi-view methods for imaging genetics. My name is Andre Altmann.
And I'm Jonas Ricciardi. So today in this symposium, of course, it's about imaging genetics. So we wanted to have a very quick introduction. Imaging genetics, as many of you know, is a set of methods to understand the link between genes or variations in genes, such as single nucleotide polymorphisms, and brain imaging. And this can be functional imaging, structural imaging, diffusion imaging, all types of imaging. And you see that on both sides of this equation, genes and brain imaging, data is fairly high dimensional. So you need a lot of data to do this type of analysis. And we're very fortunate that in recent years, we have a lot of large-scale imaging genetics data that has become available. So one of the most famous ones, of course, is UK Biobank, but also ADNI for Alzheimer's disease. The Human Connectome Project has genetics, Imagen for adolescents, and one of the large-scale projects that's been going on for a while is Enigma, which has a lot of different phenotypes and imaging and genetic data as well. So all of this data is great, but how do you analyze this and what kind of methods can you use for this?
So yes, the traditional way in analyzing this kind of data is to conduct mass univariate analysis, in which every genetic variant is tested individually against the imaging phenotype. And this produces for each imaging phenotype Manhattan plot, like on this right-hand side here. And as you can imagine, with imaging data, we have a lot of different modalities and we have a lot of different resolutions at where we can look at those images. And this can get very easily out of hand. And even though we have now large data sets, you have a massive multiple testing problem and also a loss of power in which might lead to you not identifying certain genetic imaging links. So one way forward is to use so-called multi-view approaches. And those approaches have emerged from classical statistics and are now a part of machine learning. And multi-view approaches allow you to identify pattern in one modality and link them to patterns in another modality that occur at the same time. So of course, this can be done using different imaging modalities, but we are using it now to link imaging data to genetic data.
So to explore this topic, we have invited 4 fantastic speakers today. And of course, you can probably watch these videos in any order you like, but what we recommend is the following order. So we recommend to start with a talk by Neda Jahanshad from USC. And she will be talking to us about classical univariate approaches for imaging genetics and what results they have yielded. Then we'll move on to an introduction about multi-view methods with Agoston Mihalik from UCL. And he will talk to us about canonical correlation analysis, partial least squares, reduced rank regression, and all these kind of methods, how you can apply them to your data. And also he will introduce a toolbox that they wrote.
And the following 2 talks will dive right into the application of those multi-view methods to imaging genetics analysis. And the 3rd talk will be given by Marco Lorenzi from INRIA in Sophia Antipolis. And he will introduce multi-view autoencoders and also talk about federated learning, in which you can conduct this analysis when the data is distributed across different sites. And the last talk will be by Jingyu Liu from Georgia State University, and she will introduce an ICA-based approach. You're probably familiar with ICA from analyzing resting state networks. And she will be using ICA-based approaches to link genetic data in the form of genetic variation, but also methylation data to imaging features like resting state networks.
So we thank the speakers in advance for their presentation, and we really hope that you will enjoy this virtual symposium. Have fun.
Thank you.
Hello, I'm Agoston Mihalik from UCL, and today I will talk about regularized canonical correlation analysis. In the first part of the talk, I will introduce the theory behind canonical correlation analysis, partial least squares, and reduced rank regression, or shortly CCA, BLS, and RRR. I will also discuss how they are related to each other and that they can be seen as subcases of regularized CCA. Then, in the second part of the talk, I will share some guidelines how to use these models in practice. CCA, BLS, and RRR are latent variable models. The key idea behind these models is to find associations between the latent representations of multiple views or modalities of data. In this talk, I will consider 2 data modalities, However, the models can be also extended to more. The two modalities will be represented by X and Y, where the rows of these matrices are the examples and the columns are the variables. For instance, the rows could correspond to different subjects, whilst the columns X1, X2, XP to voxel-wise measures of brain imaging data, and the columns Y1, Y2, YQ to SNP measures of genetic data. In this talk, we will discuss linear models, where the latent variable Xi can be calculated as a weighted combination of X1, X2, XP, whilst the latent variable omega can be calculated as a weighted combinations of Y1, Y2, YQ. This can be also written in a matrix form as shown on the left and on the right. The objective of latent variable models is to find these weights Wx, Wy, such that Xi and omega are maximally associated to each other. In the rest of the talk, I will use this diagram to represent latent variable models. And remember that X1, X2, XP, and Y1, Y2, YQ are the columns of the data matrices X and Y, and they refer to the original variables, whilst Xi and omega are the latent variables. The most obvious choice of latent variable model is to maximize the correlation between Xi and omega, which will be denoted by a red arrow between them. Another choice could be also taking into account the variance of one modality, which is denoted by multiple red arrows. Or we could take into account the variance in both data modalities, therefore the variances in X, Y, as well as the correlation between the latent variables could contribute to maximizing the association. It turns out that all these choices of models can be seen as eigenvalue problems, and therefore they can be solved by eigendecomposition or singular value decomposition. In this talk, I will present the solutions by singular value decomposition, or shortly SVD. All right, let's see the first latent variable model. Canonical correlation analysis, or shortly CCA, maximizes the correlation between the latent variables, subject to the constraints that the standard deviations of these latent variables are 1. Most commonly, the optimization function is written by using the covariance and the variance matrices, as can be seen now. It turns out that all latent variable models discussed in the talk can be presented in this form, such as maximizing the covariance between the latent variables and subject to different constraints. The SVD solving the CC optimization has 3 terms. In the middle, the covariance matrix between X and Y, whilst on the left and right sides, the square root of the inverse variance matrices. Although CCA looks like an appealing approach, it has a limitation in its current form, due to the inverse variance matrices marked by blue boxes. If there isn't enough number of examples, these inverse matrices will become unstable and leading to overfitting the data. Moreover, if the number of variables exceeds the number of examples, CCA becomes ill-conditioned, and these inverse matrices don't even exist. The next latent variable model is reduced rank regression, or shortly RRR. This method stems from multivariate linear regression, where the aim is to predict an output matrix Y from an input matrix X and estimate the weight matrix C. If the errors in matrix E are correlated, the solution is provided by generalized least squares, where capital gamma in the middle is a positive definite matrix. Normally, this solution assumes that the weight matrix C has full rank, which is illustrated at the bottom left. Instead, reduced rank regression decomposes C into a multiplication of A and B, which are both low rank weight matrices as illustrated at the bottom right. We can also write reduced rank regression as an optimization with constraints. It turns out that it maximizes the correlation between the latent variables, and depending on capital gamma included in the constraints, it can also maximize some of the variance in Y. Therefore, I marked the variance component with light red on the top diagram, as it's a flexible part in the model. The SVD, solving reduced rank regression, has again 3 terms. Depending on the choice of capital gamma, reduced rank regression has 2 commonly used subcases. In the first one, capital gamma is the inverse variance matrix of Y, and we see see that in this case, reduced rank regression will be equivalent to CCA. Thus, it will maximize correlation as shown in the middle diagram. In the other subcase, capital lambda is an identity matrix, when reduced rank regression will maximize not just the correlation, but also the variance in Y, as shown in the bottom diagram. The next latent variable model is partial least squares, or shortly PLS. It maximizes the variance in both data modalities, as well as the correlation between the latent variables. Maximizing these three terms is equivalent to maximizing the covariance between the latent variables, which is the common way of understanding PLS. This can be also seen from the SVD solution, which includes only the covariance term. It's important to notice that if we assume that the variance matrices of X and Y are identity, the CCA constraints will be the same as the PLS constraints. Thus, the two models will be equivalent. We will come back to this towards the end of the talk. In the following, I will demonstrate that PLS, CCA, and reduced rank regression can be all seen as subcases of regularized CCA. It was proposed in 2004 by Hardin and colleagues in this form. We see that the constraints are the combination of the CCA-like and PLS-like constraints, and they are controlled by a pair of hyperparameters, Cx and Cy, which range between 0 and 1. If these hyperparameters are both set to 0, regularized CCA will be equivalent to CCA. If they are both set to 1, regularized CCA will be equivalent to PLS. Two other corner source solutions are when one of the hyperparameters is set to 1, the other to 0. In one case, it's called orthonormalized PLS, or shortly OPLS. The other one is a subcase of reduced rank regression. And indeed, we can see that both solutions on the left represent the previously discussed reduced rank regression forms. All four corner source solutions maximize the correlation between the latent variables, and the difference of the models comes from whether they also maximize the various variance components in one or both data modalities. Therefore, similarly to before, in the diagram on the top right, the variance components are marked with light red, as they are flexible parts in the model. Now we turn into the second part of the talk, where I share some guidelines how to use these models in practice. First, we start with regularized CCA. If we write out the constraints in schematic matrix form, we can see immediately that the PLS-like constraints act as regularization on the CCA-like constraints, as they add the diagonal matrix to the variance matrices of X and Y. This type of regularization adds stability to the CCA model and is called L2 regularization, as it uses L2 norms of the weights. Another common regularization is L1 regularization, which we'll discuss in the next slide. The real value of regularized CCA is that we don't need to decide a priori if we want to use, for instance, PLS or CCA, but we can perform data-driven model selection. As all models maximize correlation between the latent variables, we can use it as a metric to compare the models. This is demonstrated in the figure on the right, where correlation is presented on the z-axis as a function of the hyperparameters on the x and y-axis. PLS, OPLS, and CCA are presented in the corners. Reduced rank regression is presented along the y-axis. We can see that in this example, the best model, which has the highest correlation and marked by a red line, is a mixture of models and very close to a PLS solution. It turns out that with high-dimensional data, PLS gives good results. Our experience working with such data is that data-driven model selection often favors PLS-like solutions, as I have just shown in the example. In this slide, I will discuss a powerful extension of PLS called sparse PLS. It uses an additional constraint, which limits the number of variables included in the model. This is because, as we see in the constraint, the sum of the weights need to be smaller than a value specified by the hyperparameters Cx and Cy. Therefore, the weights of irrelevant variables will be set to 0. It's illustrated on the diagram on the right, where only a subset of variables denoted by red arrows contribute to the associative effect. If we rewrite the constraints of sparse PLS, as shown now, we see that they correspond to L1 and L2 norm constraints, which used together are called elastic net regularization. CCA with elastic net regularization is very popular in applications. In case of high-dimensional data, the variance matrices of X and Y are often assumed to be identity. And then, as we discussed before, these assumptions make CCA equivalent to PLS. Therefore, sparse CCA and sparse PLS are often equivalent models in practice. Below, I present an example from our recent study where we applied sparse PLS in a sample of 345 adolescents, with some of them being diagnosed with depression. We used voxel-wise gray matter volumes as one modality, and item level measures of psychometrics, IQ, and demographics as the other modality. We can see that sparse PLS managed to link 7 depression-related variables to a small set of brain regions, including insula and subcortex, while selecting only about 2% of the non-imaging and 5% of the imaging variables. Therefore, we can see that sparsity facilitated the interpretability of the results greatly by selecting such a small subset of consistent variables. Finally, I present the framework we proposed in the same recent paper and discuss 2 key concepts, which are generalizability and stability. By generalizability, we mean that our main interest is not how latent variable models fits the data, but how well these models generalize out of sample. Thus, we apply the trained model on independent validation holdout sets and measure the correlation between the latent variables there. The validation sets are used in the inner loop of the framework to find the best hyperparameters, whilst the holdout sets are used in the outer loop to assess the significance of the results. By stability, we mean that our interest is to find models that are similar to each other and which they don't depend on specific characteristics, for instance, on the subsample of the data they were trained on. Therefore, we measure stability as the similarity of weights across models. On the top right, we can see the outputs of an inner loop. In this example, the X and Y axis represent the hyperparameters which control the sparsity of sparse PLS. For each pair of hyperparameters, the models are trained multiple times on different subsamples of the data. On the left figure, the Z-axis represents the average out-of-sample correlations of these models. On the right figure, the Z-axis represents the average similarity of model weights. On the left, we can see that a relatively large range of sparsity level can provide very similar generalizability. However, we can see on the right that the stability of models has a clear peak. Combining these 2 metrics, we can take advantage of both desirable properties. This is illustrated at the bottom figure, where the dots represent models with different hyperparameters. The x-axis represents the average similarity of model weights, and the y-axis represents the average out-of-sample correlation. An ideal model would have coordinates of 1, 1. Therefore, minimizing the Euclidean distance from this point will result in a model which is both stable and generalizes well out of sample. Before concluding the talk, I would like to mention that all the concepts I discussed here are implemented in a MATLAB-based toolkit, which will be released hopefully in a few months. The toolkit includes even more models than discussed in the talk, such as kernel CCA or CCA combined with PCA. It also includes various framework options, for example, using different hyperparameter selections or statistical inferences. An important feature of the toolkit is the iterative calculation of associative effects, so that, for example, we can find the optimal regularization for each associative effect. Finally, I would like to thank my colleagues for their support. In particular, Fabio Ferreira and Niels Winter, who were involved in the toolkit development, my PI Janaina Murao Miranda for the opportunity working in such an amazing environment, John Shaw Taylor for the technical help, and all members of our group. Thank you very much.
Well, thank you everyone for coming to this session and for viewing my talk. My name is Neda Jahanshad, and today, as part of the symposium, we'll be discussing functional insights from univariate genetic associations with brain structure and how we're going to use these as a benchmark for more advanced multivariate analyses. So just a quick background. You're all here in an imaging genetics symposium, but why are we studying this, or what is the goal? Basically, we want to identify some genetic variants that might be related to variations in brain structure and function, And why we want to do that is to, first of all, understand the basic biological underpinnings that might be underlying some susceptibilities to neurological or psychiatric disease risk, or that might lead to different brain changes throughout the lifespan. And the second reason is probably why we're having this symposium is because it's a difficult problem and you really need some creative mathematical and engineering solutions to be able to search this huge search space of millions of genetic variants, which could be associated or parts of tens of thousands of genes, biological pathways, and more. And there's just an unlimited number of features that can be derived from the brain images. So we'd like to use this entire search space of imaging and genetic features to be able to improve our understanding of neurobiology. So as other talks in this session focus on more multivariate and multi-view approaches to imaging genetics, and here are a few reviews, not necessarily related to the specific talks coming up, but just other reviews that might be helpful. But before we get to a lot of the multivariate analyses, we're going to test everything one at a time. So with mass univariate testing. And what we'll be going over in this session is a brain-wide, genome-wide association study, what we can do with all these univariate findings and results, and why both these univariate and multivariate methods are important. So the paper I'll be discussing today that describes the mass univariate analyses that we did is this genetic architecture of the human cerebral cortex, published in Science just a couple months ago in March 2020. What we did, we worked with over 300 researchers from around the world, and representing populations from all over the world to look for genetic variants that might influence cortical structure. We looked at global surface area, global cortical thickness, and also regions defined by the Desikan-Kiliani Atlas from Pre-Surfer, and that looked at 34 surface area, or 34 regions for both surface area and thickness. And we looked bilaterally, so we averaged both left and right and looked for the genomic relationships. So although we had a total of 70 traits, this effectively was about 60 independent traits. With approximately 1 million independent genomic loci or variants, our multiple comparisons correction threshold was p less than 8.3 times 10 to the negative 10th. So a very strict correction threshold is needed for these univariate tests. I won't get too much into the details of how we did this analysis for just to save some time. But to be very brief, we don't centralize a lot of the data. So this is done as a meta-analysis where our consortium, the Enigma consortium, sends out protocols that we put online, we make available to the community to process the imaging data, the genomic data, to do some quality control, and also to do the analytical univariate association. So the protocols go out to the community, to all interested participating sites. And then what comes back to the central group is the association summary statistics and a lot of individual level quality control metrics that we use to basically QC at the group level, find any cohorts that might be outliers for various genomic loci or imaging traits. And then we do an inverse variance-based meta-analysis on all the data that comes back. So in total, we had about 50 groups involved in the discovery results, so the principal analyses. And these are examples of 2 forest plots that you're seeing right now. With each, for every single genetic variant that was significant, we made one of these forest plots. And each row shows the finding for a particular cohort. The red shows the meta-analysis result, and we further meta-analyzed the UK Biobank data into this, which gave us our principal meta-analysis result. And then there were other cohorts that were used for replication and generalization before we determined whether our findings were significant. So just as an example, we found several genomic loci significantly associated with both global surface area and average thickness. And also we were able to map specific genetic loci to specific regions of the brain. So all this data is the data as in all the results, all the association summary statistics are available to download. So you can go to the Enigma website, click on the GWAS downloads, and request the download summary statistics. But you might ask, okay, these are just univariate results. Why would I download these? What can be done with them? The paper's already published. So I'm going to go through a few things that we did in our paper that might give someone some motivation to look further into the results. And these are just mostly to show what can be done to take the univariate results a step further. So one thing we did, you can do a genetic correlation between each pairs of regions for each of the traits and compare that to the phenotypic correlations. So in the top of these matrices, you see the phenotypic correlations between each pair of regions. And the bottom half of these matrices show the genetic correlations between each pair of regions. And you can see that they line up nicely, but don't always match. You see surface areas to the left and cortical thickness is to the right. So these can be taken further. You can do a hierarchical clustering on all the surface area results across all the regions and also all the thickness results across the regions. And you see these nice lobular patterns of clusters that show up, which is pretty interesting given that each specific region was associated with the GWAS was run independent of all neighboring regions. And you might want to know, okay, well, how do these results relate to brain function? We've done genetic correlations with several neuropsychiatric traits and disorders, and we see that at large, surface area is associated with several disorders, including insomnia, ADHD, depression, but also, so that's lower surface areas associated with higher risk for those disorders, but also higher surface areas associated with higher educational attainment and also Parkinson's disease. So this can give us some insight into the neurobiological pathways that might be associated with those disorders from a genetic standpoint. And there's a lot more insights into the biological implications of the genomic results in the paper, but please do read the paper to find out more. I'm focusing mostly on the imaging findings at this point. So how does this set of univariate findings relate to any multivariate analyses that we want to do? So you could actually sum up a lot of these individual risk scores. So you can do a polygenic risk score for prediction. So let's say if we have genetic information for an individual that was not included in the original study, can we use the results of the study to predict brain structure in any way? So how good is the full set of common variants for predicting brain structure? So we can do this by aggregating the risk for a trait by using a weighted sum of the genetic risk variance for the individual, and the weights are determined by the GWAS results. So we actually showed that in an independent sample of over 5,000 individuals, the GWAS results that we present in the paper at the univariate level sum up in a multivariate, just a linear combination can explain 2 to 3% of the variation in both total surface area and average thickness. Now, taking this a step further, we were involved so our findings were used as replication for a different study. So this is an interesting study where instead of looking at univariate associations across the entire brain and doing a GWAS of each single cortical region, instead the principal components of the surface area using these regions was determined, and a genome-wide association was performed on just 2 of the principal components. So rather than doing a genome-wide association on all 34 surface area regions individually, you can do a multivariate association with just PCA and look at do a GWAS on the first 2 components. So that was what Shin and Al did, also published just in March 2020 in Cerebral Cortex. And just this was also an international effort where several cohorts participated, and each ran PCA on their own data set, and those were aggregate and a median loading for each PC1 and PC2 were determined, and that was how that served the basis for the genome-wide associations. So you might ask, how can the univariate associations and the results be used to validate or to serve as replication for this GWAS that clearly used a multivariate approach in their imaging before running the genetic associations? So we actually consider this more of an in silico replication using genome-wide inferred statistics, or GWIS. That was paper published in 2016, which provides approximate summary statistics for an approximate GWAS that is based on a linear combination of other existing GWASs. So essentially, the GWAS of the PC was therefore estimated as a weighted sum of 34 individual regions that we had available, weighted by the PC loadings. And this showed a very nice replication of both of hits for both PC1 and PC2. So that we were able to replicate despite not originally doing a multivariate approach. There are several other studies looking at genome-wide associations in Enigma at this stage. And you, if you have the data, you're more than welcome to take part. And please do reach out to us if you are interested. We have one work on bioRxiv already by Rachel Brower, who's first author of the work, which looked at change in brain structure over time. And this change was evaluated for individuals across the lifespan, and scripts were distributed to 37 cohorts from around the world. And we determined regions that change at different points in the lifespan and the genomics that are associated with that. We have a new GWAS that will be underway shortly, so there's plenty of time to get started and be involved in that one from the start. That one would be related to white matter microstructure. So please do reach out if you'd be interested in those. And finally, I'd like to thank several of the large-scale consortia that are making their data available and available for use in these studies, including the UK Biobank, PGC, and also Charge, which has served as a replication cohort for several of Enigma studies as well. And thanks to all the Enigma analysis team for putting this together, and the over 300 individuals that were involved in this study and ran the analyses. Just a fun fact, the project started in 2014 and was published in 2020. So you can see this distributed analysis might take quite a bit of time, but the results are hopefully useful for many types of analyses in the future. Thank you very much.
Welcome to tuning in this talk. I'm going to talk about ICA-based multimodal data mining and its application on imaging genetics. My name is Jingyu Liu, come from Georgia State University. I believe everyone already motivated to do the imaging genetic studies. So here I only have one note for you. Since imaging genetics was started probably 10 years ago, it started with imaging, brain imaging and genetic data. But right now, as more and more data modalities are involved, from genetics all the way to neuropsychological assessments, including epigenetics, transcriptomics, proteomics, brain structure, and function. With such more data modality, the method to analyze them also need to be developed. Therefore, this talk is only focused on method development, precisely a set of methods based on independent component analysis, ICA. Under the envelope of ICA, I will introduce several tools. First, start with parallel ICA, then extensions of parallel ICA include reference parallel ICA and sparse parallel ICA. This set of tools analyze two modalities. Then I move to the N-way ICA and N-way ICA. These tools are designed to handle multiple, more than 2, 3, and more data modalities. Hopefully, by using one of the methods or combination of the methods, you can reach your target. So parallel ICA was first introduced about a while ago. The concept of parallel ICA is simple. What we need is apply ICA to ICAs for two modalities. One is SNP, one is brain. So when the ICA try to extract the hidden independent component, which in each data set, information among the two modalities also exchanged with the goal to optimize the correlation among them. Correlation is based on loading matrices. Loading representing how each component expressed across subjects. For example, if a genetic factor is highly loaded or expressed in one subject, then the linked brain component also needs to be highly expressed and loaded in this particular subject. mathematically, ICA is implemented within this platform. First, ICA, S equals WX. X is our observations. This is the voxels or SNPs around subjects. S is a hidden independent component. The ICA algorithm is to try to extract the W matrices, which the linear matrix can produce maximally independent S. There are different ways to get the W. The parallel ICA is based on Informax. It's based on the maximize entropy term in order to maximize independence. Therefore, for the parallel ICAs, we have two W matrices to update. W1 for dataset 1, W2 for dataset 2. And W matrices will be updated based on the entropy term. And secondly, W matrix will be maximized based on correlation term. Correlation is based on correlations A1 and A2. A are loading matrices, representing how each subjects are loaded in, how each component are loaded in each subject. Therefore, when the algorithm converge, we will get a W matrix, maximize independence, and also optimize the correlation. In practice, when parallel ICA was applied in the practical problems, there are several items to consider. The first, number of variables, number of voxels, and number of SNPs. And generally, ICA can reliably extract the whole brain voxels. For number of SNP will be relative to number of sample size. Based on our simulation and our experience, if you have about roughly say 200 samples, then you probably can analyze reliably less than 10,000 SNPs. The last thing to consider is component number. In general, the component number embedded within each data set are different. So we will use estimation. We'll estimate the component number for each data set separately. For brain imaging data, we generally use information theory-based estimation, like MDL, AIC, to estimate number component. For SNP data, which are slightly different, because the genetic component, each genetic component carries smaller variance. From largest component to smallest component, the variance are continuous change. There's no clear turning point. So we use stability-based methods to estimate the components. We already built the tools embedded in the toolbox. Then after you estimate the component number, you can apply to parallel ICA to your data set. Parallel ICA has been applied to multiple projects. Here, I'm just using one example to demonstrate the feasibility or utility of the parallel ICA. This project, we applied parallel ICA to schizophrenia cohort. In discovery sample, we have 1,777 samples. In replication sample, we have 600 replication samples. We select 1,402 SNPs based on the PGC schizophrenia GWAS results. These are top risk SNP after pruning. Then we apply the method to whole brain voxels. In this project, we identify one paired brain component with SNP component. And this link are replicated as consistent significant in discovery and replication. Brain component focused on the frontal parietal junction area. In this region, the green matter volume is significantly reduced in schizophrenia patient cohort. And these components are linked to SNPs within chromosome 6 P22 regions. We did several extra steps to verify the results. First, we did compute project risk score for these 30-some SNPs based on the PGC GWAS results. And turns out our genetic components is very similar, almost the same to the project risk score computed based on PGC results. Therefore, this indicates with much smaller data set, by data-driven approach, we can identify genetic component which carry some meaningful results derived from large population studies. We also did additional tests using postmortem brain tissues transcriptome profiles and postmortem tissues DNA methylation profiles. To verify risk alleles we identified for reduction of the green matter are same risk alleles for the transcriptome alteration, DNA methylation alteration in postmortem cohort, in postmortem tissue of a schizophrenia patient. And the extension of the parallel ICA is parallel ICA with references. There are two reasons we build these tools. First, for some projects, we do know prior knowledge. We do know which genes, which SNPs should be involved, should be highlighted. Second, when we process a large amount of SNP loci, for example, 500,000 SNPs, if we simply apply the ICA on the data, the component we identified carry large variance may not be the component which interest for this particular project. Therefore, we build references. Reference means that we highlight the regions we know that should be highlighted, should be involved. For the rest of the regions in the cross-genome, need to be using data-driven approach to define whether it's involved or not. So in other words, we're looking at the components has highly weighted in this particularly reference regions. Mathematically, we build a distance measure between the references and the component, and we want to minimize the distance between the reference and the components. The advantage of this approach is we can analyze much larger number of SNPs. In this particular example we showed here, we analyzed the 500,000 SNPs. And the second advantage is this method can handle multiple references. If you have a set of genes, which you don't know whether the genes should interact or how they interact, you can list off the genes at the multiple rows of references. Algorithm will automatically to matching the references, to combine the references in the data-driven way. Another extension of parallel ICA is sparse parallel ICA. The reason for this, also, we know the reason for designing this algorithm is that we know the SNPs across genome, only a small set are interested for particular projects. So we add additional sparsity control on the components. We implemented nonlinear Fourier transformation based on the ratio of L2 and L1 ratio. L1, L2 norm ratio. The advantage of this approach is that the component we derived has a much enhanced signal strength and suppressed background. Indirectly, this method also increases the number of variables, number of SNPs to be analyzed, and also increase the replicability. We apply sparse parallel ICA to real data on ADHD cohort. In this study, we have 341 adults as discovery. We have 461 adolescents data as a replication. We select 2018 loci based on the ADHD GWAS results. Now apply the sparse parallel ICA to whole brain voxels. In discovery's data, we identify one SNP component, which highest peak region in the chromosome 5 long non-coding RNA regions, interacting with several genes. Some of the genes has already reported a relationship with ADHD, and this gene are related to brain components. And in replication, we replicate one pair of connections. This genetic component are consistently related to the gray matters in middle frontal and superior frontal regions. And the connection is much stronger in case families than control families. Now we move to the N-way parallel ICAs. The rationale to design this approach is that in current practice, it's very common to collect a multiple data set, like structural MRI, functional MRI, your DTI, or genetics or epigenetics. So we want to have a method to analyze the data simultaneously. So we designed this N-way parallel ICA using 3 data sets as example. For data set 1, 2, 3, we have 3 ICAs in parallel. And each ICA is trying to identify components within each data set. In the process, we will match the loading matrices, loading A1, A2, and A3. We were exclusively to pair the top paired component. And then we will optimize sequentially each paired components. In practice, because the convergence issued, this algorithm works reliably for the top paired components. Therefore, the advantage of using this method is can simultaneously analyze multiple modalities and can reliably to extract the top paired related components. And just this year, we developed a new method, Anyway ICA. The rationale to design this new method is for some conditions, the paired component are multiple. There are multiple paired components exist across data modalities. So we designed this method to simultaneously to optimize the all the top paired components. Using 3 datasets as example, we have 3 ICAs to conduct it. Each ICA extract the components and also loading matrices. And then instead of exclusively sequentially to optimize these components, we build a subspace component vectors. Subspace component vector, number of vectors depends on minimum number of components. Use this example. We have dataset 1 has 5 components, dataset 2 has 4 components, and dataset 3 has 6 components. So therefore, we have 4 subspace vectors. And then we will minimize mutual information across the subspace, component subspace. Minimize mutual information indirectly maximizes the covariance of which within each subspace. Therefore, advantage of this algorithm is we can simultaneously analyze n modalities, and also we optimize entire loading matrices via mutual information. At the same time, we optimize multiple paired components. So in summary, I introduced a set of tools-based ICA to perform multimodal multivariate analysis. This set of tools is still growing. We're still developing new methods to meet the challenge of evolving the new data sets. Hopefully, by this talk, you can know that if you have some certain multimodal multivariate analysis projects, ICA, this set of tools may be one of your options to analyze. Last but not least is I want to use this chance to give thanks to my lab members and collaborators. Dr. Chen is involved in reference ICA, Dr. Victor Vergara and Dr. David Botti is involved in the ICA, and Dr. Lin is involved in transcriptome the DNA methylation studies, and Kwai Kwai Duan is my student who is involved anyway ICA. Thanks for your attention.
Okay, good morning. My name is Marco Lorenzi. I'm a researcher of the PM research group at the area Sofia Antipolis and Université Côte d'Azur. And my presentation is about a latent variable model for the analysis of heterogeneity in biomedical data. And from an analytical perspective, analyzing heterogeneity means modeling the variability that we can observe in biomedical data, for example, imaging and genetics data. And for modeling this variability, we have to make assumption about the nature of the signal that we measure concerning this large array of imaging and genetic information. And so, of course, the signal is representing some anatomical or biological process, but also it can be affected by several factors, for example, bias due to acquisition or processing, and then noise. And ultimately, we would like to understand, so to model the joint variability of this kind of signal. And actually, we would like to depict a causal direct association, for example, how genetics influences the phenotype we see in the image. But we must be aware that most of the time we see associations which are mediated by other factors which are many times unknown. And this association can be even spurious, so it can be due completely to random factors. And due to the complexity of this kind of problem, most of the time we don't have a valid a priori hypothesis or general a priori hypothesis in this kind of association, so everything has to be inferred completely from the data. And the problem becomes even more complex because when we want to gather very large data samples, we need to analyze data that comes from multiple centers, and this presents other issues, other sources of variability due to population, acquisition, processing again. And also we are confronted with the problem of data security, because most of the times this data cannot be shared, and so algorithms must be deployed in such a way that they don't require to centralize data in a unique server. In this presentation, I would like to propose and show how latent variable models can be used to tackle all these kind of research scenarios of analysis problem, starting from the analysis of multivariate signal with an application in imaging genetics. And so latent variable models can be actually explained through a generative formulation. So basically, we assume that our data, as complex as it can be, is generated from some simple latent representation. This latent representation here is denoted by the variable z, which is a low-dimensional variable, so something that lives in a simplified space, that then can be transformed by opportune transformation, here f and g, that can actually create or reconstruct our data from z. So knowing f and g means that we know perfectly how these different data views vary together. And then we can make additional hypotheses on how the data can be corrupted, for example, by bias, noise, or adding further actually factors that can modulate our signal to provide a complete description of the data. So knowing this generative process, we know perfectly, we have a complete description of all the variability of the data, can be used for analysis, for inferring features and characteristics related to pathologies and so on. And ultimately, in our analysis, we want to solve the inference problem, so inferring all the parameters of this generative model, given the data. Classically, this problem has been formulated as a linear problem in which the latent variable is linearly transformed into the data space through projection matrices, projection vector. And so it requires to solve jointly this joint multivariate linear regression model for the inference of both latent variable and projection. And it can be shown actually that solving this problem amounts actually to modeling the cross covariance between the different view x and y's. And this motivates an alternative formulation of this kind of problem, for example, in classical approaches like PLS and CCA, in which actually we aim at finding the best optimal projection of each data view in a respective latent space, in such a way that the latent space is, so the respective latent space have a maximal association. And this association can be measured, for example, in terms of statistical correlation, and here we have a CCA, canonical correlation analysis, or can be measured in terms of maximal covariance, and here we have partial least squares. So there are ways of deploying this method to make them scale to high dimensions, and here we can see, for example, an application of partial least square that we propose for the analysis of imaging genetics data from a cohort from the Alzheimer's disease neuroimaging initiatives. And so you can see on the left-hand side, so concerning the brain features, the colored areas are the ones that have been depicted by the model for being the most significant for explaining this association. And you can see the model identifies regions like temporal, temporoparietal region of the brain, which are known to be involved in Alzheimer's disease. Things become more complex when we look at the genetic data, because you can see here the parameters of the model along the entire genome from chromosome 1 to 22. And although the model is able to depict, for example, relevant location, for example, ApoE genes in chromosome 19, well, there is much more, and actually this, what the model is really useful is in pointing actually relevant location, so significant location, and it can be used in downstream analysis. For example, by using this location to query gene annotation and expression databases. And so with our analysis, we show that from the insights from the PLS model, we could identify a low number of candidate genes that then could have been tested to identify actually promising genes to explain the pathophysiology of Alzheimer's disease. One of the frontiers of latent variable model is the extension to what is called multi-view setup, in which we don't want just to explain the variation between two different views, but have a complete explanation of how different kind of view, for example, imaging or genetic, clinical, biological data vary altogether. And again, here the problem is essentially a problem of scalability, because we have to deal with a very high number of dimensions. And also a problem of flexibility, because most of the time a linear transformation might not be enough due to the complexity of this kind of relationship. So still we can try to formulate latent variable model, borrowing some of the concept and the tools from a modern machine learning concept. For example, the concept of autoencoders, in which we have again our latent transformation, and we want actually to find transformation, so a decoder that explains the data, so maximizing the probability of the data knowing this latent representation. And since solving this inference problem usually is very challenging and is prohibitive from a computational point of view, what it has been proposed to do is actually to infer an approximation of this latent representation from the data itself through an operation which is called decoding. And depending on the view that we consider, we can have different decodings, so different approximations. And ultimately, what we would like is to have them actually be coherent and actually represent the same latent object. And this can be imposed, and what we propose is to impose a constraint actually in how these latent representations are inferred. For example, impose that they must be, for each view, they must be as close as possible to the same common target, which would be the ideal posterior distributions. And we can show that optimizing these constraints amounts at optimizing another quantity, which is called evidence lower bound, which is a very interesting quantity, because when we optimize through this elbow, basically what we are doing is we are finding an optimal encoding, so an optimal representation for a given view, which at the same time, it can explain all the other views. And we would like to this optimization to be carried jointly across all the views, so to enhance the consistency of this representation. Plus, this formula presents also a regularization term that we impose to be like a model selection terms to favor simple models. So the simplest latent representation, and here is enforced through a sparsity inducing prior through dropout. And here you can see, for example, an application of this model for the multi-view analysis of multimodal imaging information and social demographic factors, clinical score. And you can see that the model is able to identify a consistent latent representation that is clinically meaningful because it discriminates across different clinical categories. But also from this latent representation, we can sample and generate actually variation, so different prediction of, for example, imaging features to analyze how they all vary jointly through actually the pathological axis that we identify in the latent space. So this provides a very powerful way of analyzing and understanding the variability that has been modeled by the approach. So the model actually is open source, and there are currently investigations using the model. And I would like actually to share with you a couple of ongoing projects beyond actually the field of neuroimaging. For example, trying to predict the visual fields from retinal nerve fiber layer thickness map in ophthalmology, here on the left-hand side. Or in cardiology, actually try to predict an entire CMR images, cardiac images from the fundus image of the eye. So this points to the flexibility and the scalability of this model for actually these very challenging applications. And last, I would like to show you a further extension of this kind of approaches for the challenging problem of multicentric analysis, in which actually the first challenge we have to face is the problem of data access, because most of the time the data cannot leave the centers, and so it has to be analyzed in a meta-analysis setting. And while meta-analysis as a discipline, statistical discipline, is quite a developed discipline when it comes to standard statistical testing, for example, meta-analysis of sample size, p-values, when we actually deal with complex models like latent variable models, there are many challenges we have to face. And already a few years ago, we investigated how, for example, a partially square model, which relies on the estimation of the cross-covariance matrix between the different views, how this kind of model can be actually distributed across different centers without sharing the data. The idea is simple. We have to estimate an eigencon decomposition of the cross-covariance matrix, and if the covariance matrix or the data matrix is partitioned across centers, actually this is to show that it can be factorized as the sum of the center-specific covariance matrix. So we can just share covariance matrices, or even better, approximation of covariance matrices to actually reconstruct a global partially square model. And this motivates also the use of different schemes, for example, for sharing information across center to have this global latent variable model estimate. And of course, research is moving, models are becoming more and more complex, and currently we are trying to scale actually this kind of approach. For example, the multi-channel variational encoder, in general, like more complex deep models, to the multi-centric case. For example, our current project Fed Biomed is a multi-centric analysis of imaging genetics data in neurodegeneration, with a focus on Alzheimer's and Parkinson's disease. And actually here we are currently studying a new paradigm for flexible and scalable approaches of these complex models by leveraging on federated learning. So to conclude, I hope I convinced you that latent variable models are powerful theoretical and provide a powerful theoretical setup, and also are actually very powerful tools for analyzing variability in heterogeneous and complex data. And these days, they become more and more exploitable thanks to the advances in machine learning, for example, through stochastic gradient descent, backpropagation, and so on. And usually, there are a lot of parameters involved, so we need to gather a lot of data, and that's where we see that federated learning can be actually a promising solution for further developing the use of this model in practical scenarios. And of course, from the research point of view, efforts are needed to integrate even more hypotheses about well, the bias and noise, so better modeling this heterogeneity, and introduce also a priori knowledge about anatomical or biological structure of the data to include in the model. So if you're interested in the topics that I showed in this presentation, at my webpage you can find more material, composed by tutorial, workshop, and codes, and so on. And I would like to thank all the collaborators, in particular the ones that contributed to the results that I showed you in this presentation, and all the members of my team. And thank you very much.